Major depressive disorder is a chronic psychiatric condition that causes changes in moods, thoughts, behavior, and physical health. The hallmark of major depressive disorder is the occurrence of dysphoria and anhedonia for a duration of at least 2 weeks. The underlying pathophysiology of major depressive disorder is yet to be clearly defined. Even though there are numerous studies attempting to shed light on the pathophysiology of depression, it still remains elusive. There are numerous hypotheses about the physiological underpinnings of MDD. This table presents just a few of the hypotheses surrounding MDD, including genetics, deficiency of monoamines, and impaired circadian rhythm. Transcranial direct current stimulation, or tDCS, is a non-invasive and safe neuromodulation technique in which a weak electrical current is applied to the cerebral cortex via 2 scalp electrodes. This is seen to result in lasting changes in neuronal excitability in underlying cortical regions depending on the polarity of the stimulation. More specifically, anodal tDCS typically leads to an excitatory effect through neuronal depolarization, while cathodal tDCS sees reduced neuronal firing resultant to hyperpolarization. It has been shown that tDCS changes cortical excitability by modulating the membrane resting potential, especially during the period of stimulation. Grimm and colleagues developed the hypothesis that the left dorsolateral prefrontal cortex is hypoactive in depression. Thus, current studies using tDCS attempt to modify activity in the prefrontal cortex. Specifically, they attempt to enhance excitability of the left prefrontal cortex and reduce excitability of the right prefrontal cortex. Numerous meta-analyses have followed tDCS and its effect on MDD. In 2011, a review of tDCS by Nietzsche and Paulus found the effect of tDCS on depressive symptoms to be mixed. Many studies found tDCS to have an antidepressant effect, while others found it had no effect whatsoever. Another meta-analysis conducted by Kulu and colleagues in 2012 also found similar results. In 2014, a meta-analysis conducted by Shizawa and colleagues investigated the efficacy of tDCS as a treatment for MDD. This time, the study found active tDCS to be statistically superior to sham tDCS in the treatment of MDD, suggesting our knowledge and understanding of tDCS on major depressive disorder must be improving. Despite these promising results, there are still a lot of unknowns in the world of tDCS, specifically regarding its effect on MDD. First, future studies need to explore various aspects related to the efficacy of tDCS. Currently, it is not clear which brain areas should be directly targeted with tDCS to achieve optimal antidepressant efficacy. Additionally, variables such as the length of time stimulation should be applied and how many sessions are needed to achieve stable effects all need to be investigated. Also, alternative electrode sizes, shapes, and placements should be looked into. Second, further studies should also investigate if higher stimulus of tDCS is safe and tolerable, and also a more effective treatment. Third, larger studies and studies in different populations are needed to confirm the results of previous findings. Lastly, long-term follow-ups are needed to evaluate the stability of the effects longer than for 4 weeks after stimulation. Overall, our understanding of the physiological effects of tDCS on major depressive disorder has improved significantly over the past decade or so. However, it is clear there is still so much more to be discovered.